Okay, we're going to do a short, brief lecture on protein synthesis. Okay, so how your cell makes very important proteins. It's going to be based on the synthesis or production of those proteins. And that's going to be done by the assimilation of amino acids that will eventually be joined by peptide bonds in order to create our protein. Now, there's three phases that we want to look at, and the first of which is transcription, the second of which is RNA processing, and then the last, which is the translation, which will be performed by the ribosome. And that's what's going to take us from DNA to RNA and then eventually our protein. So if we look at this little diagram, it shows us how we take DNA, unwind it, and then produce our pre-messenger RNA, which, because it's going to contain some regions that are not considered to be coding regions, and so it'll have to be processed. We'll go through RNA processing in order to remove those regions in order to produce our functional RNA, messenger RNA, and then finally that messenger RNA will leave the green region, which is our nucleus, and move into the purple area, which is our cytoplasm, where a ribosome will attach in order to produce our protein. Before making proteins, we must first make RNA. So how does RNA and DNA actually differ? If I can grab a pen right quick. The base of RNA that's going to be different is going to be uracil. And so the base in DNA will be thymine. The other bases are that you find in both are guanine, that's an I, cytosine, and adenine. Okay, as far as the sugars, the sugar that you'll find in RNA is going to be ribose. The sugar that we'll find in DNA will be deoxyribose, hence where we get the names ribonucleic acid for RNA and deoxyribonucleic acid for DNA. And both of those are 5-carbon sugars. For RNA, we'll have a single strand, and for DNA, we'll have a double strand or double helix. Okay, get to the next slide. Okay, so then transcription, which was the first of the process that we talked about out of the three phases, where we take DNA and transcribe it into messenger RNA. DNA will be copied into a complementary sequence of messenger RNA. And remember again that instead of using thymine in your RNA molecules, we will only use uracil. And then the other three that we mentioned. The other molecule that's going to be responsible for promoting transcription, and that's going to be RNA polymerase, along with other transcription factors. So if we look at this drawing, it's showing that RNA polymerase, along with the other transcription factors, are going to be responsible for helping to unwind small sections of DNA at a time in order to produce that pre-messenger RNA strand. Okay. So a question is, what would be the complementary RNA strand for the following DNA sequence? Because now, if I can grab a pen again, remember we said that we would not use thymine in our messenger RNA. So let's say if we're creating our messenger RNA, and then so G would pair with cytosine, or guanine would pair with cytosine, cytosine would pair with guanine, guanine would pair with cytosine, thymine would pair with adenine, adenine in DNA would pair with thymine, but this is RNA, so that's going to be uracil, and then thymine will pair with adenine, and guanine will pair with cytosine. So just wanted to make sure that you are capable of making a strand of RNA. Okay, so the types of RNA, there are 3 types of RNA. The first is messenger RNA. Remember, messenger RNA is the molecule that's going to be produced as a result of transcription. The molecule that's going to be responsible for reading the messenger RNA or reading the codons is going to be the ribosomal RNA. So it's going to translate that messenger RNA into a sequence of amino acids. So who's going to be responsible for transferring each amino acid? That's going to be our transfer RNA. So it's going to have an anticodon, which will tell us, based on the codon, which of the amino acids is going to be transferred into the sites located on the ribosome. Okay, and remember that all of these molecules are produced in the nucleus. So if we look at messenger RNA strand that's produced, one of the important parts of the messenger RNA is going to be that start codon, because that's going to tell us where we're going to start this translation process. And then eventually we're going to have also a stop codon, which is going to determine the termination of that linear sequence of amino acids, which are going to eventually make that protein. So again, the messenger RNA is carrying instructions from DNA to the ribosome in order for us to produce essentially every single protein that you'll find in the body. This is showing us our ribosome. If you notice that the ribosome is composed of 2 subunits, a small subunit and a large subunit. And if I could grab a pen, I'll underline that. So here's our small unit. Or maybe I didn't grab a pen. Okay. Okay, so here's the small subunit. Here's our large subunit, the biggest blue part. And then you'll also see that there's what we call a translation groove. And the translation groove is where the messenger RNA will be fed so that we can read each of the 3 bases, which stands for the codon on messenger RNA. So again, this is our translation groove. You also notice that there are 2 sites on the ribosome. The P site, that is the site where the initial tRNA, which is going to initiate the process of translation, will be attached to, so it'll bring in the first tRNA, which will be carrying our first amino acid, which will be methionine. The second site is where elongation will take place, where the transfer RNA will start to bring in other amino acids based on the code that's found on messenger RNA. So based on that codon that's in messenger RNA. So we'll read every 3 bases, and we'll move from one. After we've read one, then we'll keep moving down until we end up meeting that termination sequence. Okay, so if we can move on. Okay, and then again, the transfer RNA, this is the guy that we refer to as our go-getter. It's going to be responsible for going out and picking up the right amino acids that are available in the cytoplasm according to the message that's on the messenger RNA. So according to the codon. So those 3 bases is what's going to tell us which of the molecules that we're actually going to go out and pick up. So which amino acid. At the top you'll see our amino acid attachment site. I'll use a black pen, but here we're talking about where that amino acid will actually be attached. And you'll notice down at the bottom the anticodon area, which will match the codon or complementary base pair with the codon that's on messenger RNA. Okay, and that's, remember we said AUG was the start codon, and this is the tRNA, which is going to be the initial or the initiator tRNA, which is going to get this process of translation started at the ribosome. Okay, so then with RNA processing, remember we said once we've produced our messenger RNA, that the messenger RNA will have to be processed. It is a pre-messenger RNA molecule that has coding regions as well as non-coding regions, also called nonsense regions, and we call those nonsense regions introns. And those introns will actually have to be removed in order for us to produce a functional messenger RNA. Once we remove the introns, the exons will be joined together and form our functional messenger RNA. On the next slide, I'm going to show you the entity that's going to be responsible for helping to remove the introns, and those are going to be your spliceosomes. So here's showing you the pre-messenger RNA molecule, and again, the lavender areas are the exons. The grayish purple areas are your introns. That's the nonsense material that has to be removed in order to produce our functional RNA. So the spliceosomes are going to be responsible for helping to remove these regions so that we can produce our functional or mature RNA, messenger RNA molecule, which will be sent out into the cytoplasm so that it can be joined with the ribosomes. Alright, so translation, what are we talking about? Translation means making protein. So there are 3 parts that we need to look at, and essentially those are initiation. So we have a start codon, which is the AUG region of the messenger RNA molecule that's going to tell us where we're going to begin this process. And remember, the start codon is going to be somewhere near that P site that you saw on the ribosome. Remember, there was a P site, there's an A site, and then the A site is generally where elongation will begin. That's where the proper transfer RNA will be brought in to, so eventually during elongation, a peptide bond can be formed between that methionine that was brought in by the initiator transfer molecule, and then this second one brought into the A site of the ribosome, and a covalent bond is formed between those two, and eventually we're going to move down every 3 bases until we reach our termination sequence, which is our stop codon, which is something like on the messenger RNA, the UAG, or we may see UGA, or we may see UAA. Those are some of the other stop codons. Okay, so let's make a protein. Okay, so again, this is just showing you that whole process again. But here, this is our ribosome, and you'll see the P site. Remember, we said that the messenger RNA is going to line itself up with that P site near where? The start codon, which was the AUG. So here, down at the bottom, you'll see, and I'm going to try to grab a pen, but you'll see that the AUG of the messenger RNA codon is lined up at the P site on the ribosome. Okay, and this is where that first initial transfer RNA will bring in our first amino acid. Okay, and just to, before we begin to look at the universal genetic code chart, if you remember the 20 common amino acids, that's what these codons are giving us. So these codons, UUU, for example, is telling us, see, can I grab a pen just so I can do a little underlining here, that will produce phenylalanine, or CCU tells us that will produce a proline, or for example, the AGA says that we're going to produce arginine. So you get a gist of how this chart works. So we're going to be looking at, again, the codons every 3 bases found on which molecule. Is it tRNA? Is it messenger RNA? Okay, right, it's on messenger RNA. So that's what we're going to be looking for in order to figure out which amino acid is going to be next in the chart. So let's go down here. Remember, we said AUG was our start codon. Okay, alright. So if you look at initiation, you'll see that our amino acid number 1 is being brought in. The anticodon, which is complementary base pairing with the codon in transfer RNA, is bringing in that first amino acid. We said the first amino acid, something like methionine. And remember, where is that first amino acid lining up? It's going to be right in the middle, and that's at our P site. Actually, if I grab a pen, and that's where eventually the release would take place, but there would also be an E site over here, okay? And that's where the transfer, the first initial transfer RNA is going to move over into the E site once this, the one that's in the A site has been bound, so that we can move along, and then this is where that transfer RNA will be released. The E site, and move over to the E site, then actually leave, okay, or be released from the molecule. Alright, let's keep going. So then you see that the transfer RNA number 2 is coming in to bind where? In the A site. So this is, if I can grab a pen and just label these real quickly. Okay, so we said that this is the P site, and then this is the A site. So the second transfer RNA is coming in. Now this is where elongation, when this second transfer RNA comes in and binds to the proper codon, then this is when elongation begins. Because remember we said we start with initiation, and we had our initiator transfer RNA come in, and then we want to go to elongation. And eventually, after we've added all of the amino acids that were to be coded for, we'll finally come to the end where we'll find our termination sequence or termination codon. Okay, so let's continue on. So then you see where that binds. The other elongation is the formation of that covalent bond that we call a peptide bond. And I'm going to try to circle or underline that for you. So here is the peptide bond that's being formed between amino acid number 1 and amino acid number 2. The next thing that's going to happen is the transfer RNA in the initial spot will move over to the E site and eventually be released. Elongation is what we said was happening here. Okay, so here comes transfer RNA number 3. And you notice transfer RNA number 3 comes in. Transfer RNA moves to the E site and then is eventually ratcheted or removed. Okay, so then that's going to mean that the amino acid number 2, which was in the A site, will have to move over to the P site so that we can move amino acid number 3 into the A site. Because remember, elongation is taking place at the A site. So here comes amino acid number 3, and then there we go. And we'll keep doing this same thing until we complete it. And notice between each one of these, part of elongation is what? Again, the formation of the peptide bonds between the amino acids, because what we're doing is creating our linear sequence of amino acids, which is going to lead to the primary structure of a protein. Okay, so again, amino acid number 3 will move into the P site, and amino acid number 4 will eventually come in as we move this ribosome down the messenger RNA. It will move into the A site. So number 4 will move into the A site. So let's go on. Okay, so we see 4 coming in. Okay, here we go. Until we've got finally elongated till we look, now we're up to amino acid number 200, and then we see what? Our terminator or stop codon. We finally reach our terminator or stop codon. Okay, UAG. So remember, if you look on page 103 in your textbook and look at what UAG says, it says it's a stop codon. Okay, so this is going to terminate that protein's synthesis process. So that's termination. So then our end product is what? The primary structure of a protein. So what have we done? We've taken a sequence of amino acids and joined them together by peptide bonds in order to produce that primary structure of a protein. And in the next video that's coming up, you'll see that some of the interactions that take place within that primary structure, especially the side chains, will lead us into secondary structure, and then finally into tertiary structure. And then when we have more than one of those tertiary structures bounded together, usually by some type of cofactor, will produce quaternary structure. But we'll get to that a little bit later. But anyway, here you see again our protein. And so my final question, all right, the anticodon, UAC, belongs to transfer RNA. Okay, the anticodon, UAC, belongs to transfer RNA that recognizes and binds to a particular amino acid. Okay, it says, well, so let's work our way back. It says, what would be the DNA base code for this amino acid? So if tRNA, we said, was UAC. Now, let me see, can I grab a pen? So, was UAC. And we said that was our anticodon. So the complementary base paired to that would be A, U, G. Okay, remember that was our start codon. If you looked at that chart on page 103. So then the question asks, well, what was the DNA, okay, segment? What was the DNA segment? So let's grab one more pen. So DNA would have to be what? T, A, C. So that was our original triplet that we saw. That was the original. All right, so finally, in this, that we've taken DNA from DNA to our protein. So we started with DNA. We made our messenger RNA, and from the messenger RNA, we created transfer RNA brought in our amino acids. The amino acids were joined together by peptide bonds through first initiation, elongation, and then eventually our termination sequence, and so that we finally yielded the primary structure of a protein, which is its linear sequence. We'd like to thank you very much, and I hope you got something out of this quick lecture. See you next time.